Hey everybody, it's been a really long time since I've been on a webinar. It's kind of a little weird to not actually see some faces lined up on the side, but yeah, we'll roll on and see how everything goes. As David said, Elizabeth Garofalo, UMass Extension Fruit Team. Tree fruit pathology is my primary focus, integrated pest management specialty. And of course, I do dabble in insects when I have to. But I don't touch weeds. Thankfully, we've recently acquired a weed management specialist at UMass. So, should you find yourself with weed issues, um, let me know, or you can reach out directly to Maria Gannett and for those things. But for today, we're going to be talking about some disease issues. We're going to go over some fire blight, bitter rot, apple scab, peach leaf curl, brown rot. We'll do some of the biology, some ID, look at some lookalikes for those diseases that have them, and some management issues or questions. Fire blight, of course, we know and loathe this particular disease. Erwinia amylavra is the pathogen that causes it. It overwinters in these cankers that you see here on this trunk. You can see it's sort of discolored, the bark is cracking, and then you can see that this branch here has also become diseased as a result of that canker. So in the spring, when temperatures get to be roughly around 65 degrees, those cankers, the margins will become less distinct and they'll look a little water soaked. And that's when that bacteria is really moving around through the tree. So eventually that bacteria will start to ooze out of the canker. And that's when we have the ability to spread the inoculum. Now, of course, we need to have these open flowers in order for our initial infections to occur. And so what happens is we can have either rain splash dispersal from the cankers or insects will crawl around on the cankers because that ooze is kind of sweet. So it will attract insects to feed on that. And so they get their sticky little feet in the ooze, which has the bacteria on it, and then they'll fly over to the flowers. And then that bacteria gets on the stigma of the flower. And so once it's there, it will reproduce. And then with a wetting event, all of that bacterial population will get washed down into the nectaries of the flower where it can enter into the host through those openings. So then what we get is this primary infection here. You can see in this red circle, there's a flower cluster that was infected. And then what it did, the bacteria got into the shoot, gummed up all of the vascular tissue and cut off flow through the shoot. And so that's really what causes the crooking of the shoot here, which you can see this characteristic shepherd's crook, as we call it, on the shoot here. So that's our primary infection. But then we get a little further into the season after blossom is over, and we have these, again, you see this flower cluster here that got infected. And in this particular instance, the shoot was short enough that the infection went directly into the trunk, and you can see this canker starting to form here. So this infection court here, the shoots and then the trunk here, those are all going to be active infections. And so those all have the ability to ooze further bacteria. Not only can they ooze further bacteria, but that's going to move through the tree, and that's going to lead to what we call shoot blight. So there's blossom blight, that's our primary infection, and the shoot blight typically comes up as a result of that primary infection. And that will continue to infect other shoots, and it has the ability to also get into fruit that have formed that weren't actually infected through blossom infections. So we can also see that infection over here with this little apple that's starting to get all gooey and gross. So the ooze isn't always white, it isn't always brown, but typically if you're seeing anything that's oozing, it's going to be fire blight. In apple, that is. Um, and so, of course, these then create cankers that overwinter, and that's our next year's inoculum. We have some management options. More and more, we're seeing research showing that some biologicals have efficacy. And so this is sort of my interpretation of Carrot Cox's and Anna Wallace's fire blight management protocol. And they have it starting in obviously in the dormant season. And so removing those cankers in the winter is a critical management strategy. Getting rid of all of that potential bacterial inoculum is really going to help reduce your pressure moving forward. We then move into our dormant to delayed dormant copper applications. And what this does is it knocks back the population of bacteria that's existing sort of on the surface of things. So if those canker margins have started to soften and the bacteria is moving around and coming out, that copper application is really going to knock that population back and sort of set you up to be a little bit cleaner to begin with. Then we move into this pink time frame. So we're still not talking about strep yet. At this pink time frame, we're really looking at putting on our systemic acquired resistance inducers or our induced systemic resistance materials. So basically, whichever one of them you're dealing with, one of them is stimulates leaf tissue and the pathways, genetic pathways within that. Another one stimulates genetic pathways through the root system. The end result is largely the same in that what it does is it kicks the plant's immune system on and tells the plant, hey, we've got something going on. You need to start putting out your antibodies or whatever it is that you're using to protect yourself from an infection. So we want to do those at pink because it takes a little while for them to kick on. So then, of course, we move into bloom time, and we really want to manage, first of all, using our forecast models, right? So NUA or RIMPRO, whichever one you use, AgRadar may be coming back sometime soon out of UMaine. But you starting to look at those before bloom happens, because if you wait until if you wait too long, you might find yourself in a situation where you're looking back at an infection that happened and potentially not getting the management that you need. So you want to start looking at the forecast model a few days in advance of when you think you're going to be dealing with bloom. And then it's also, of course, it's really important to put your actual bloom date into the forecast model, because while the forecast model itself will sort of estimate what the bloom date is going to be, it's really not going to be the most specific to your location unless you put your date in. That's, of course, when we start to think about our strep. And so that still remains the most effective management practice for blossom blight of fire blight. Then we do want to go back in again after bloom with some more of those resistance inducers. And then this just helps moving forward and helps reduce shoot blight issues. You can use Apogee for that period as well, but there is a trade-off there. So if you're trying to get your trees that are new up to that top wire, that Apogee is going to slow that growth. But the slowing of the growth is also the thing that's going to slow the bacterium. So there's really sort of a trade-off going on there in what your biggest concern is, whether it's fire blight or growth. So again, this is from Anna Wallace and Carrot Cox, just a list of materials that they have looked at that includes this. Obviously, streptomycin outright kills the bacterium. We have a handful of materials that are what I'm currently calling a blocker. And you'll notice the numbers along here. These numbers correlate with the slide that we were just looking at, and that's that number management by the numbers. So 5s, 3s, 6. These are those numbers just indicate where you slip it into your management program. And then we have these blockers. These aren't those systemic resistance materials I was talking about. These are basically going to occupy the space within the floral tissue that the Erwinia would otherwise occupy. So it's like a land grab kind of thing. So they're taking up all of this space. Some of them take up space and resources, and then they prevent the Erwinia from being able to either land and reproduce, which will, of course, then minimize the inoculum buildup that you get into the tree itself. The other ones down here are the resistance inducers that we've already talked about. These antibiotic metabolites, these are biocontrols that produce something that is akin to an antibiotic, sort of like strep, just not as effective. And it does do the same thing, and it outright will kill some of the pathogenic organisms. So one material that is not currently, I don't have on this particular slide that also falls into that systemic acquired resistance material is Actigard. It's not organic the way some of these others are, but it is effective and another tool in that toolbox. Moving on to lookalikes. So fire blight, because it can be so bad because it can really cause so much damage. Typically, when we see a shoot that looks like this one over here, the first response from anyone, myself included, is to think, oh boy, we've got fire blight. However, that's not always the case. And with this particular instance, what we have here is we're dealing with Nectria. And so Nectria is a pretty weak pathogen. Typically, I've only ever seen it in Rome beauty. But it gets into these pruning cuts, right? You see here, this was cut off here, and then this shoot here grew. Well, so this is where that infection got into the shoot itself. It does cause a little bit of the shepherd's crooking, but that's not because of anything moving through the vasculature. What is happening here is this fungal pathogen has basically strangled off the base of the shoot, which when it comes to causing the shepherd's crook, the end result remains the same. But the good thing about this is that it's not bacterial and therefore it's not moving through the vasculature of the plant. So it's not great, but chances are it's going to be a one-off strike in a tree, and it's not going to move through the rest of the tree the way fire blight will do that. Another lookalike for fire blight is the Pseudomonas syringae. I'm looking at that now and I misspelled it, so sorry about that. But you get the gist. And so this is also bacterial. Unlike fire blight, which requires warm temperatures, the Pseudomonas is happier under cooler temperatures. I've only, in my 10 seasons with Extension, seen this 1 year. And it was a cool, wet spring. And again, it was one of those things that looked like fire blight. So we went out to figure out what it was, but it just didn't quite track. First of all, primarily, of course, because of the temperatures. It was just not warm enough. But then you look at it and it's, yeah, kind of doing the shepherd's crook thing, but not really the way you would expect it to with fire blight. But it does look a little bit similar. And then you can see this over here. And again, it gets into the tissues and it causes death, but it typically is not a problematic pathogen for apple. Okay, so moving on to bitter rot. I can only imagine that you are seeing, like the growers in Massachusetts, an increase in bitter rot incidence and severity. It is, so you are going to have to listen to me use this terrible, terrible joke and pun for the next however many years that you have to deal with me. It's a complex problem. And I say that, and it's a terrible pun, I'm sorry. I say that because the causal organisms, it's not just one. There are multiple complexes within this Choletotricum genus that can cause bitter rot of apple. Now, in the Northeast, we typically believe that we are dealing exclusively with the Choletotricum acutatum complex. Further south, where it's a little bit warmer, they are dealing with the Choletotricum gliosporoides complex. The gliosporoides complex is the one that causes that glomerella leaf spot. And since we typically don't see that here, that's kind of what is leading us to believe that we're dealing primarily with the acutatum complex. Within the acutatum complex, there's a number of different species that can cause this apple bitter rot. We have a bit of a life cycle here. And so in the field, so there is theoretically a sexual aspect of the Colostotricum genus. It's an ascomycete, just like apple scab is, and so it has the potential to reproduce in a similar fashion. However, it's not observed, the sexual aspect of it is not observed in the field. And that's important because the sexual aspect of it would be the spores are shot, right? Think about the way apple scab shoots its spores. And so if this were to be found in the field, it would have the ability to shoot its inoculum further than the spores that are asexual, because the asexual spores are splash dispersed or potentially thrown around, carried around by insects as well. And so that's really the aspect that we're dealing with over here. So we have overwintering on mummies in the tree. There is some evidence to suggest that this pathogen, it doesn't infect fire blight strikes, but the spore can live in the dead tissue caused by a fire blight strike. So one more reason to get rid of those fire blight strikes. But so it overwinters as mummies. And the spore, the inoculum really is kind of there throughout the season, but it does have a point at which it becomes greater. There's higher pressure later on. And so then the mummies will release their spore. The spore will cause infection. Now, that infection has the ability to lay dormant for a while. So we could have an infection theoretically at the end of May, let's say, but not see any symptoms develop up until we get into July, right? That's because that infection is just, it's not necessarily seeing the conditions that it wants to in order for it to develop further spore. And that's eventually what it will do. So that first infection will lay dormant, then eventually it's going to kick on. It's going to start to decay the fruit, and it's going to spit out, it's going to push out fruiting bodies, and those fruiting bodies are going to then spit out spore. Those spore will then splash disperse within the canopy onto clean fruit, infecting those fruit. And so one of the challenges with bitter rot is that it doesn't require any wounding. It can infect whole sound fruit. So then those infections continue to go through the season, and then those mummies or those apples that are infected become mummies. They stay in the tree, and then next year there's your source of inoculum already there, ready to go. So we have seen both the Gliosporides and the Acutatum complex here in the Northeast. As I mentioned before, we're operating under the some research has shown that Acutatum is around. Not a lot of that research, however, has been done actually in New England. And so when I say we're operating under the assumption, it's a stronger knowledge than assumption, but we haven't really rigorously pinned down which species we're dealing with. However, I will say that this summer, myself and Dr. Nate Westrick at the Connecticut Ag Experiment Station were able to identify Colostotricum fiorinae causing bitter rot in apple in Massachusetts. So we're working on getting those species identified. And we need to identify those species because they have different fungicide sensitivities. And so if we find ourselves with something in the Gliosporides complex, we might be having a different fungicide program than if we're dealing exclusively with Acutatum complex species. As I mentioned before, they're rain splashed within the canopy, so the dispersal is limited as compared to what those ascospores, that sexual phase, would look like. And the primary inoculum does come from within the canopy. All of the growth stages of apple are susceptible, but that inoculum load of spore increases as the season wears on. So that late dormancy to fruit set is where that spore density gets really high. And then, of course, that will continue on through harvest. So Surgeon Chimovich is a extension professor at Virginia Tech. He recently gave a talk for another online series. And this is the fungicide program that he sort of laid out as if he were going to deal with bitter rot based on the research that he has done, which if you're interested in looking at the research trials and the different fungicide trials he has done, this YouTube link would take you there. I will share that with David and Katie later in case you folks would like to go see that. And so here's basically what we have. The first two, three covers are primarily directed at apple scab, right? So that's really what you're dealing with right now. And the other thing that's happening is remember that inoculum load for bitter rot is not super high at that point. And so in blue, that highlighted in blue right there, that's considered to be sufficient for bitter rot because you're really not dealing with bitter rot. Captan has some efficacy, especially Captan plus Prophyte does have efficacy against bitter rot under low pressure situations. So this is a good way to start things off. Then we're going to move into our group sevens. So it's really important to rotate, remember? And so this is laid out, surgeon color coded this to lay it out in a way that makes it really easy to see the rotational options. And so one of the things that he has done here and laid this out is he's front loaded the program, those fourth cover roughly and on, with the PHI, longer day PHIs. So you've got your Approvia, and that's a 30-day PHI. He's suggesting 2 applications of that before you rotate in with a Flint, which is a FRAC 11. The Approvia is a FRAC 7. We also have Omega in the mix now, which is a relatively new FRAC. It's a group FRAC 29. And this is one of the things that our growers here in Massachusetts have been using. I don't know, maybe you folks are using it as well, but we're finding good efficacy. It is a little on the more expensive side because it's new. And so hopefully over time that will drop down, but it is an effective tool against bitter rot, especially if you're really struggling with that. So then after you have your 2 Omega, then we switch to the lower PHI options. So we've got our Pristines, Maravon, and Captan. And so we're mixing Captan in with all of this because we don't, like, it's just another one of those resistance prevention mechanisms. So one thing to keep in mind is your later ripening cultivars are going to require continuous coverage. If you don't keep them covered, if you have bitter rot and you don't keep them covered, they are going to develop bitter rot either in the field or later in storage. And then that disease is just, again, it's going to continue to spread throughout, it'll spread through your bins. So sticking with the surgeon playbook here for another slide. If infection does get started, remember, so with disease, it's really, especially with bitter rot, it's strictly a preventative situation. Bitter rot does not have a curative fungicide. It's because it gets into the tissue. Once it's there, it's there. And so if you do find that you're seeing symptoms, especially on your more valuable cultivars, you're really going to have to stay on top of it. And so one of the things that over the past years we have suggested is, stretch out those dry periods. We're not saying that so much with bitter rot the way we might with apple scab. If you get a 2-inch rain, if you get 2 inches of rain, you really need to go in and reapply and not wait for the 14-day stretch. Going in with a full rate of Captan plus Pristine with the Maravon, Luna Sensation with LI 700 gets really good fruit coverage. So then you're keeping that fruit really well protected. And of course, the 10 to 14-day intervals without the rain. So I mentioned that the inoculum overwinters in the mummies in the canopy, right? So getting those mummies out of the canopy is a really important way of reducing your disease pressure. Even during the growing season, and again, I realize that there's a labor and time component to these suggestions, but if you are able to get the fruit that are actively infected through the growing season out of the canopy, and at the very least, get it into those grassy drive rows, you will help break down that fruit and remove the inoculum from your clean fruit, right? So, and especially in a high value cultivar like Honeycrisp, if you find yourself seeing bitter rot in your Honeycrisp, it might be worth your time to take out the symptomatic fruit to help protect the fruit that's clean in there. Opening up the canopy to ensure that your airflow is good. And as I mentioned before, the fire blight and any other dead wood has the ability to harbor spore from bitter rot. So no mummies. There are a couple of lookalikes for bitter rot. So we saw this picture earlier just to kind of drive home. Bitter rot has a couple of distinctive features, one of which is the concentric sporulating salmon-colored rings. This is those fruiting bodies that I talked about earlier that have burst forth from the tissue and they're spitting out their spore. And their spore is these gelatinous little pustules, if you will, of inoculum. When you cut into it, you typically will see this cone-shaped progression of rot towards the center of the apple. And you can see here too, along the side of the apple on the outside, the rot lesion is flat. Whereas with your white rot, it stays round. And earlier on, this is a particularly advanced example of white rot, but earlier on, you can actually see white discoloration around the lesions of the rot. And then the fruit itself stays round. Black rot, you can see some like concentric ringing here, but you're not going to see that pink sporulation with black rot. Also, it goes straight, the rot goes straight through the apple. So that's some ways to determine whether or not you're dealing with bitter rot versus white or black. Apple scab, I'm not going to spend too terribly much time talking about the life cycle of apple scab. I feel like folks probably are pretty familiar with that. Just as a reminder, it overwinters in the leaf litter on the orchard floor. So any infections that got started last year on leaves like this, those are currently primed and ready to go to cause infection for this year. And that's important to keep in mind when, given that last year, again, at least in Massachusetts, and I know in New York as well, there had been such a significant outbreak of scab. Dealing with that inoculum is really going to be important to reducing your pressure this year. We don't want to see another outbreak. So in getting on top of that sanitation as best you can is going to be important. We do have a new material coming out for this year from UPL. It's called Axios. It's a new frac. So that gives us a whole new rotational material for apple scab. I do not believe that Axios is currently labeled for bitter rot. However, people are looking at it in terms of its efficacy for bitter rot, and we'll be doing some of that work in lab assays as well. So more to be said on Axios as it relates to bitter rot, but right now it's a new tool for apple scab. So sanitation this time of year can be challenging just for the mud alone. However, I really feel like of all of the years, if you are going to do sanitation, this year is the year that it's the most worth it. Leaf chopping at this time of year, if nothing else, you're going to help speed up the decay, but you're also going to flip the leaves. So the way that apple scab grows in the leaf is the fruiting bodies will grow with their ejection port facing upwards. So no matter how the leaf lays in the fall, come spring, the ejection door, if you will, is facing upwards. So if this time of year you come along and you flail up those leaves, you're going to be breaking them down, making them easier to decompose, but you're also going to be flipping some of that over. And so what that's going to do is it's going to it's going to point that ejection port down. And so the spore, when it ejects, will just go to the ground and it's not going to cause any issues. So if at all possible, get in the orchard and get your sanitation done. And the other thing too is that delayed dormant copper will give you some protection for really early green tip apple scab infections. You're going to want to look at your green tip date and get for your earliest scab susceptible cultivar. Um, Typically, folks look at Mac, but if you have something else that's like ambrosia, I know got hit last year with scab. If so, whatever your earliest cultivar is, that's the green tip date you're going to want. And like we were talking about bloom earlier, the decision support system will estimate a green tip date for you, but again, it's not going to be as accurate as your eyes on your trees and then putting in your own date. So, recently, so Carrot Cox and his lab out in Cornell have been monitoring dodine resistance in apple scab populations, and they have not seen any evidence of resistance even in their resistant research blocks. And so he's actually suggesting that this is now a tool that we can bring back into the rotation, and especially relevant now that we are potentially looking at some manzate and Captan further restrictions on those materials. So he's suggesting 2 applications are acceptable. Don't do more than that. We don't want to lose it again. And then, of course, saving your single sites for later in the season and tank mix them with a broad spectrum so that we can continue to have those materials and have them be effective. So those are going to be your FRAC 7s, 11s. Sivia, of course, is a good material. That one also helps with powdery mildew. And then again, Axios is that new material that we were just talking about. Peach leaf curl. Overwinters in buds of trees that have been infected, we saw a bit of an outbreak of this last year. I think, on the heels, typically on the heels of a year where we don't have a crop, we tend to see peach leaf curl because people aren't really thinking about disease management. So right now, you may, depending on your site-specific situation, be able to get a material on for peach leaf curl. In the past, it has been just recommended that we use copper, but now that we're seeing it become more prevalent, we're also suggesting the use of a chlorothalonil like Bravo. Something to be said about that. So I'm not sure how far into the discussions around the Endangered Species Act and the EPA's new website folks have gotten. We at UMass are just sort of starting to get our heads around this ourselves. However, chlorothalonil is going to be the first fungicide that I'm aware of that will have this ESA language on the new labels. So keep your eyes on that. Try to start to get to familiar. And I've included the link here, which I will send to David and Katie for you all. The website's a little onerous right now. Not a shock. But depending on what your environmental conditions geographically are, you might have endangered species at certain times of the year. And so with this whole website, this bulletin's live too. You go in there and you basically you put in your site, you put in your material and your spray date, and you have to show that you have done that and confirmed that there is no endangered species that will be impacted by your application. The herbicide module has already launched, and so that's something else to keep in mind. Again, I won't get into that. That would be something to talk to a weed specialist about. So, right. So overwinters in the buds, as those buds start to swell, the spore that is already there is going to cause infection. This does not repeat infection throughout the season. So, once you see it, don't spray it. It's not going to do you any good. You want to get it either in the fall with your chlorothalonil and or copper, or early in the spring with that as well. Otherwise, you're just wasting material. Okay, so brown rot. The pathogen itself is another one of those that theoretically has a sexual stage, but we just typically just don't see it. We don't have the conditions here in the Northeast in order to cause the fruiting bodies, these apothecia here, to develop. And so for us, what we see is overwintering in mummies and cankers right here. And then these mummies, they're going to produce our conidia. That's our asexual spore. Those are then going to go and infect blossoms. And from there, that infection is going to travel down through the blossom and has the potential to create new cankers on the twigs that the blossoms are attached to. So that's our blossom blight for brown rot. So blossom is our first time for management. Then those blossoms and cankers hang out there, and they, of course, are going to produce further spore, but also those mummies that are there from last year are also going to continue to produce, put out spore. So our next time is really looking at our fruit ripening. So typically, hang on. So yeah, so the temperature is really important. 50 degrees Fahrenheit, we need about 18 hours of wetness. 77 degrees Fahrenheit, we need about 5 hours of wetness. So that's something to keep in mind if you're looking at bloom and wondering if you need to worry about brown rot at this point or not. If we have these conditions, then, and you've had brown rot in the past, then you do have the potential for infection there. And then 2 to 3 weeks prior to harvest, as that fruit starts to get, the sugar starts to increase in the fruit, then it becomes susceptible as well. We typically, again, another one of those materials that you have to prevent. There's no kickback, or pathogens rather, excuse me. There's just no kickback on this. Recently, I saw George Sundin from Michigan State University present on he's a pathologist from Michigan State, present on stone fruit disease management, and they are seeing resistance to Indar in Michigan, which is concerning because it's a highly effective material for brown rot. So that's something to keep in mind. I'm not aware of resistance here, but it means that if you are going to use Indar in your program, it's even more important to make sure that you rotate effectively to maintain the efficacy of the material. Typically, green fruit isn't susceptible to infection by brown rot unless we have an issue with a like stink bug or some sort of piercing sucking insect of that nature. If they puncture the fruit, then there's a way for the spore to get in there and then cause infection of that green fruit. But typically we don't see it until later in the season. Mummies, again, removing the mummies from the canopy is a really important way to reduce your infection. And then, of course, if you can, pruning out cankers in the wood as well. There are a number of different materials that you can use. So I have this right here. So Miravis would not be a good rotational partner with either Maravon or Luna Sensation. Remember, with those premixes, you have to be extra careful about what you rotate with so that you're not rotating one of the materials that's already in that premix. And then these single sites, mix them with a Captan or a microthiol disperse in order to, again, work with that resistance management.